Okay, hello. We are group number 5, so we'll talk about influence of geometry and topological constraints on genome organization. So let me remind you briefly what we learned about the first week. So the genes are organized in a diverse fashion depending on the scale. And we now have insight into the architecture of genome thanks to high-C experiments. And the usual output of those experiments are contact maps, which for each pair ij of points on in the genome give us the contact probability. And then a thing which is easier to look at is this profile P of S, which is simply summed over all the points or the pairs of points which have the same distance S. And then this profile is quite robust and quite reproducible and has this feature of a power law with a slope -1. And our idea, the idea of the project was to try to recover some of the features of P of S with a very simple model where we model the polymer with self-avoiding random walk in different geometries and different obstacles and try to find what's the scaling of P of S. In particular, we know that in this, let's say, mesoscopic region, we get this dense packing where P goes like S2 -1. That corresponds to extensive scaling of volume with length of polymer, in turn subdiffusive scaling of the distance with the genomic distance. So we'll try to recover that using different geometries and constraints.
Okay, in order to simulate the configuration of the chromosome, we have tried different walks. The first one is the original random walk, which an agent chooses the nearby position randomly. The second is the myopic self-avoiding walk, which an agent only chooses the nearby unoccupied positions. And because of the ability of sensing the nearby positions, the myopic self-avoiding walk might generate some bias. In order to get rid of that, we've tried several ways to change the configuration of the self-avoiding walk. The top two are the local moves, where only very few positions are involved in making those moves. And the popping is the only way to change the length of the trajectory. The bottom two are some kind of global moves, which can involve a lot of points. And by doing just a few global moves, we can sort of like change the configuration hugely. And by doing this, we can have the walks and the trajectories, and we analyze those trajectories.
So as we learned, first we have to study the most boring and useless case, like free self-avoiding random walk in unconstrained geometry. In this case, we can measure P of S and R of S, and what we find is that they scale as they are supposed to scale. So it's boring. It's excellent. Then before we start playing with something like super complicated, we can ask like what is the effect of the constrained space. So we say that our like self-avoiding random walk can walk only inside the box, and for different boxes we can measure P of S and R of S. So basically we can scan everything from isotropic space that would correspond to a cube. And if we change ratios of our box, we can go to effective two-dimensional space and even one-dimensional space. However, we find that it's not enough to constrain our walk to get that -1 exponent, so we have to do something else. Also, one may examine different contact maps, and one can notice that in case of simply constrained random walk, contact maps has no feature. So we have like just simply power law decay. But we can see some kind of So basically, what we expect to see is that like we have like regular decay and then some plateau, and in some cases we indeed saw decay and plateau. That's good that our intuition is in agreement with our observation. However, we definitely need to do more to get highly packed random walk, and next person will elaborate on that.
Okay, so in order to explore other ways to constrain this DNA chain, we tried just cubic boxes with obstacles in them, hard obstacles. And in what I'm going to explain, these are all myopic self-avoiding walks. And in exploring this, we considered two scenarios. The first one, a bit inspired in biology, in the fact that inside the nucleus, these chromosomes sort of have their own territories, also as there was at the introduction. And what we do here is we first generate some walk and use this one as an obstacle for, say, a second walk, so adding a second chromosome here. And we can do this sequentially, adding more chromosomes, if this ever goes. Yeah. Up until, for example, 6. Became very computationally demanding here, so we only tried up to 6. But from here you can, for example, see how the Rij looks like. And just to remind you, this is just a matrix that says the distance between a point I to a point J here. And so we see that in including just one chromosome or several packed ones, there are different structures, say, in these matrices. However, when you do several of these runs and average all of these sort of runs, you get an Rij that doesn't really have any structure. It's just thick here on the diagonal, which is expected from just an unconstrained random walk. Moreover, if you look at the R of S and the P of S, what we obtain are the scaling exponents expected for just a myopic random walk. So yeah, and including more chromosomes there's not really a difference in any of these curves. So it's like it doesn't make a difference to add these obstacles this way. The second scenario with obstacles was to include rods in the box that extend the whole height of the box here on different densities. So like really a dense mesh of rods going to less rods. Again, we did some random walks on these ones, and if we look at the results, again we find that the exponents are those of just a random walk, just a self-avoiding walk, and again there are not many differences in how dense or not this array of rods is. So not really a lot to say here, but in the next part it will be more interesting, hopefully.
Okay. So far, everyone has talked about obstacles in the random walk and how they would constrain the random walk. And we've seen that it doesn't change so much, except for the plateau thing. So it's known that DNA is actually constrained topologically. So we wanted to investigate this with our random walks, self-avoiding random walks. So the idea is that a long polymer chain can be seen as a loop, all right, locally at least. So we're going to simulate loops, self-avoiding loops. So the loop has a topology in itself. It can be knotted or have multiple knots. And loops can be also entangled. So it has a topology with respect to its neighbors. And this is the idea of a topology constraint. Either it's constrained by its own topology or by its neighbors. And so our idea was to generate a self-avoiding loop and to grow it in a network of constraining neighbors. So the idea we had is generate a self-avoiding chain and close it using a pivot move. So the pivot algorithm is what has been described before. So this closing provides us a loop that is self-avoiding and also that we expect that describes correctly the ensemble of self-avoiding loops, right? It's not biased, less biased. So doing so, we indeed have a loop. And this loop, we observe that it behaves as an equilibrium globule. meaning that it scales with the usual exponents, all right, without the plateau here because it's not confined in space. And so what we want to describe next is the situation where we have constraints, and this situation is the following. It's like the fractal globule where the polymer actually collapses onto itself at all scales and is not entangled locally, meaning that you have some blobs that are self-repulsive in some sense. So this scales with s minus 1 and s to the power 1/3, and this is what we try to investigate later.
Okay, so we have seen that with open chain in a confined box, we don't get, we get the usual self-avoiding walk scaling with a loop, but in an unconfined space, we also get the usual self-avoiding walk scaling. So how can we get this super packed -1 scaling? Maybe you already have an idea, which is basically we have a loop within a box. Right? So how do we do that is, well, we also have rods, that's just obstacles. So we start from the left, which is, start from a small loop, we grow it with the local moves that we discussed earlier to the desired length. So here you can see that this chain is going around the obstacles, but for example, if we pull on the loop, we can pull it away, so it will not be entangled on the obstacles. You can see here already it's kind of space filling, so let's check the exponent. What we found is actually indeed we have, for R of S, we have the exponent that is pretty close to 1/3. In the beginning it doesn't really matter, it kind of scale as the SAW, but then as we increase the length, you really see the 1/3, and also it's not a finite size effect because we can increase our length of polymer 8-fold and we still see it. So similarly, we can see in the plot of P of S, we see some exponent that is -0.8. That's really close to -1. Okay. So what we want to do next is first maybe to explore the loops that start from a non-trivial topology, so maybe with some knots in it. We also need to better understand the contact maps. We haven't been showing it that much. The reason is for the same geometry and the same constraint. It's kind of hard to because there isn't like a sequence as in DNA to actually align it and take the average. So a lot of features actually get smoothed out if we take the average. So we just need to figure out a smarter way of doing it. And eventually we will hope to reconstruct the 3D structure from the content map. Okay, so we'll actually thank Ilya and Leonid for all their help and all the discussion with all of you. Thank you.
What would be the roads in real biological system?
Okay, it's a very tricky question because the model is a bit, roads just, if you go back to the globule here. Yeah. So when I say that it's topologically constrained, it is that each little globule here, now I have a pointer, here or here, all right, cannot interpenetrate with its neighbors. So in some sense, a road would be one of these neighbors in this picture we draw, all right? So if it's very complicated to explain this, but if I look at this, here, this polymer has a length, a thickness, and the thickness would be here, this thickness, for instance. All right, so it's at one scale. So at every scale, the polymer is constrained topologically by the neighbors it has here. So, it's every rod is like a pile of different chains in some sense. All right. So we simulate this as rods because being entangled means not getting the rod inside the loop. It's not very easy to understand.
I was wondering what actually happens if you have several self-avoiding loops and you let them run at the same time. So because you did it sequentially. Oh.
Yeah, so far what we did was the open chain, like many open chain, and I think it would be really interesting to see, for example, if we have multiple loops, even just to run a sequentially, what will happen. I suspect, like loops will act as obstacles to each other, and we'll probably see similar effects.
Simulating multiple loops at the same time is it's a bit the same as putting the rods. Some people have actually done it, and what you observe is indeed, as you increase the size of the loops, you see this n to the power 1/3. So the loops, they are not entangled, so it's like topology constrained in the same sense.
Yeah, just to add on that, actually in one paper they did do like chains all together at the same time. It was more like molecular dynamic simulations, but they also see that they sort of don't care about each other and they just go on entangling to each other without really like forming these compact structures.
Yeah, but then if you use like many loops and do it, you kind of see the segregation of chromosome.
Any other questions?
I already asked. Maybe you can go.
How would you go about trying to reconstruct the actual 3D structure from the contact map? I mean, could you, since you have, you actually have the structure, you simulate it, could you just pass this off to a network and do loads of simulations, get the contact map, and then you have both ends? Or am I kind of...
Yes, it's still work in progress for future.
Well, you have a nice idea.
Thank you for the question.
Could you go back to your last result? Yeah. So for RS, I'm not sure whether that's actually a power law. It looks very curvy to me. Could you justify?
Yeah, I agree. We probably need like much longer length and then to see what happens. Yeah, also it's probably the geometry also affected. So just need to do more work and then see what happens.
That thanks the group.
Hello, we're going to present the project Bits and Pieces, originally titled Entropy Reduction by Timing Selection. First, let's give a little bit of background to understand the project and the motivation of why we work on that. So the T cells are a kind of white blood cells, belong to a kind of white blood cells called lymphocytes. They play a very important role in the immune response. They start as immature cells in the bone marrow, then they move to the thymus, where they develop. Actually, that's why they are called T cells. Then out of the thymus, they continue to differentiate and they go through clonal expansion. They can be distinguished from other T cells because they have a receptor in the surface of the cell called the TCR or T cell receptor. We can see here that this receptor is able to detect different antigens. However, as you can imagine, there are like a huge amount of antigens and the body needs to be able to detect them. If we try to encode this huge diversity of immune receptor genes, which is between 10 to the 8 to 10 to the 10, then the DNA wouldn't be able to fit inside the nucleus of the cell. How does the immune system solve that problem? It stochastically generates a lot of different receptors in a process called BDJ recombination. Here, so the genome has some templates called B, D, and J. When we have these DNA sequences, one of each of these templates is taken. This is a combinatoric process. It generates around 10 to the 3 different possibilities. However, it's not enough. So here in the junctions, some nucleotides are eliminated and some others are inserted in a non-templated way. And it's actually this process what gives the diversity that we need. However, Of course, if you have a completely stochastic process, you're going to generate receptors that do not bind to anything, or actually bind too strong with self-peptides. If they don't interact with anything, they get discarded in a positive selection. Around 80% of immature cells get discarded there. Then if they bind too strongly with self-peptides, they can trigger an autoimmune response, an autoimmune disease. So they get removed by negative selection. So you can think by now that you have an initial distribution generated, but then after this process of selection, the diversity is different. So we want to characterize how that happened. There's a good model for the diversity pre-selection. There's a software for that called Olga. And for post-selection, we actually look at sequences obtained from high throughput sequencing. We actually look at an entire DNA sequence, the nucleotide sequence of the receptors. We took unique sequences because we don't really care. I mean, we know that there's clonal expansion after they leave the thymus. Right now, we don't care about that. We just care like what's the effect of the thymus selection. So we filter by unique T-cell receptor sequences. And then we focus on this region, the CDR3, that all you need to know is that it's a very important region to detect antigens. And we convert this to amino acids, which is the biological important part. So, okay, now we have the sequences pre-selection and post-selection. What do we do with them?
Okay, so given the complex developmental processes that T cells undergo, we'd like to understand how the body regulates their function. And a large part of the question just reduces to understanding and delineating exactly by how much the thymic selection process tunes the repertoire diversity. And so the overarching question that we want to address is, can we quantify the effect of thymic selection on T cell receptor diversity? And we quantify this change by measuring the change in the entropy of the ensembles before and after thymic selection. And ideally we want to do it for both the individual scale and the population level scale. So now that we have set up the question, we can think about what sort of approaches we'd like to use to address the question. And one approach would be to model the process as a noisy filtering procedure. That is, we build a model and we fit the parameters to the data that we have and use the model to estimate the entropy. And this is exactly what Alex, Thierry and team did in their PNAS 2014 paper. A second approach would be to use a generative model. That is, we feed data into a generative model and in principle we have an infinite amount of data and we estimate the entropy using this model. However, this clearly has a pitfall in the sense that the generative model is only as good as the data that we put into it, and so well, there are issues because the data that we have is fairly sparse. And the third approach is using a model independent estimate. That is, we directly look at the data and estimate the entropy from it. And this is the approach that we'll be using. Okay, so let's also look at what the data looks like. So typical sequence lengths range between 8 to 20 amino acids. And for a single individual, we have order of 10 power 5 sequences. So given what we know about entropy, let's try thinking about what we would normally do. The most naive estimate would be to tabulate the frequencies of each individual sequence and use the summation p log p formula. And clearly this approach is bound to fail because the total number of possible sequences that we can have for a amino acid sequence of length n grows exponentially. And so the number of samples that we need to make reasonable estimates of each individual, of the frequency of each individual sequence also grows pretty rapidly. And so given this issue, we'd like to pivot our strategy to placing bounds on how much the entropy reduces, and we do so by using an independent site model. And we also use more sophisticated estimators of entropy that converge a lot more rapidly even if we have less data. So more on it by Honlong.
Okay, so so the next most naive thing that one can do is to compute the entropies directly from how do you press? Sorry. Directly from the frequencies at each site. This gives us an upper bound because we are ignoring correlations between sites, which would always reduce the entropy. So because different T-cell sequences have different lengths, we first spend quite a long time thinking about how best to align them. But because of some issues that I'm about to tell you, in the end we decided to divide the T-cell sequences up by length, compute the amino acid entropies per site separately, and then average them together later to find the sequence entropies for a given patient. So for example, here is one patient restricted to T-cell sequences of length 15. We see that the entropies at sites near the beginning and near the end of the sequences are pretty low, and this reflects that these sites are pretty conserved, and also is the reason why we had trouble basically doing basic naive alignments like left aligning them or right aligning or stuff like that. So to convert from these one-site entropies to the entropy of sequences, consider the following. Consider a sequence of length L. We consider the probability of it being generated to be given by the probability of observing a sequence of length L multiplied by a product of the one-site entropies of that sequence, given that the total length is L. So if you do the math, you find that the entropy of the entire sequence repertoire has two terms. One is the sum of the one-site entropies restricted to a given length, which we have on the right plot here, and we sum them up weighted by the probability of observing each length, and there is also an entropy term due to the length distribution, which is on the left here. And so so this is basically the first maybe semi-working method for computing the sequence entropies. So what can we do with it? The first thing we did was basically to apply this to a data set containing a cohort of around 700 patients. And so this is shown on this slide. We found basically that the sequence entropies for different patients are distributed around some value of around 35 bits. And if we apply the same method to the pre-selection distribution, we find an entropy reduction of around 5 bits. And perhaps if we believe these kinds of estimates, maybe we can perhaps say find some kind of biological correlate between different parts of this distribution.
Okay, so then the next question you might have is, can we quantify how good that independent site model was? So the first thing you might think to do is calculate the entropy for a given pair of sites. So for example, site 1 and site 2 together. Calculate that entropy and compare it to the entropy you would get from individual sites 1 and 2. And if you take that ratio, if that ratio is roughly 1, that means our independent site model was good. And if it's less than 1, that means that there's some kind of correlation between those pairs. So what you can see from this plot here, this is the ratio of the entropies that I'm plotting right there, for this given length sequence that we're selecting by, which is 15, you can see between sites 5 and 10, our independent site model is actually fairly good. But in the beginning of the sequence and at the end of the sequence, there's actually some correlations between sites, which is kind of what you would expect because those are the conserved regions. So then the next most obvious thing to do is you would basically use this exact same estimate but rather than using independent sites, you're using independent pairs. So the only thing that changes in this whole calculation is now, rather than your alpha bit size for one site being 20, it's now 20 times 20, so 400. And you can then recalculate all your probabilities per pair, sum them up in the same way. And so, for example, for just this 15, you get an independent pair entropy of 30.7, which is a reduction of about 2.5 bits compared to the independent site model. And then, of course, you'll have to actually do that for all possible lengths that you find in your distribution. And if you do that again, you're going to get a reduction of about 2.6 bits. So then you could imagine going beyond pairs to triples, to quadruples, etc. And so what I'm plotting here is in red the entropy estimate for one first single site, then pairs, then triples, then quadruples, versus just the sum of independent sites, all starting at the same site. And the question is, how can we trust this estimator? And the red is a special, it's a better estimator than just the naive estimator. But what you see is if you subsample the data, it actually is a bit of a biased estimator. So it's kind of, I would say you could maybe go up to 4 or so. You just have to a little bit worry about that bias. So in summary, the naive estimator was bad because of undersampling. Our independent site model provided an upper bound of around 35 bits post selection, which is 5 bits of entropy reduction. So basically, it reduced the total sequences by a factor of 32. In the future, one of the other things I didn't mention about the n-tuple, you could even go back to the pair model, and instead of just doing nearest neighbors, you could allow it to go between any pair of things. So in general, ij, and then any partition into relative pairs, and then you can calculate the entropy of various partitions and find the entropy that's the smallest as another bound. And then you could do that for higher order things. Another thing that you could do is you could look at, treat in more detail the clonal sizes. And I want to just thank the organizers of the summer school, and specifically I would like to thank the professors we talked to during this project, Thierry, Alex, and Ilya. Thanks.
We're all getting tired, right?
You talked about how it was impractical to just like figure out the frequency of every sequence because they're just too long. But you did that with tuples of length 10. I mean, that seems like that might also be prohibitively long. Like, how much data did you have?
For each patient, we had around 100,000. So.
Oh, okay.
It's just not. It's not enough. We're in a severely undersampled regime when you get to that long. I just was plotting it to give an example.
Sorry.
I have really basic question. Maybe you covered it, but what does single bit mean in terms of like T-cell selection? I guess I'm just not used to this framework of using info theory and
I guess one bit of
imaging.
reduction is like a factor of 2 in terms of
Factor
diversity.
to what?
Yeah, entropy reduction would be like maybe if you look at the post-selection distribution, it's like a factor of 2 less diverse than the pre-selection one. So that sort of tells you the operation of thymic selection.
Okay. Oh, sorry.
Oh, could you maybe repeat the last point again, like about you show that there's n-tuple case and like 1/4, 1/8, one like that part? Yeah, like what?
Yeah, so
what were you?
this
saying?
was it was kind of just to look at how good the estimator we were using for estimating the entropy of these groupings was, and the estimator we're using was the NSB estimator, which is basically an estimator that looks at coincidences in the data to basically say something about the entropy. And we're just trying to see how good that estimator was as we subsample the data. So does the estimator scale with the data? And what you can see is as you subsample, the estimator does change its value for the very long sequences.
So what you're saying is that since your data itself is really small, that the gap between your like blue and red one, it could be due to that.
It could be due just to undersampling errors. Yeah.
Okay, let's thank this group. And group 1, I guess, right?
Hello, hello.
Testing, testing.
Okay, so we'll be talking about phenotypic selection with fluctuating environment. So as we know that the environment is often very stochastic. So if you want to deal with the environment, one of the evolutionary strategies that has developed is called bet hedging. So basically the idea is don't put all of your eggs in one basket, right? So to give an example, if you look at annual plants in deserts, what they do is they release a lot of seeds, but these seeds don't necessarily germinate in the next year. Instead, they'll go into the soil and they'll come out 2 or 3 or many years later. And these are just different species over here. And the idea behind that is if you have a bad year, then you're not going to lose all of your seeds. And you can see, in fact, that the mean seed survival rate is actually higher for species who have a low germination fraction. But in many cases, the environment is stochastic on a time scale much shorter than the generational time scale. So you might also want to switch your expression, your phenotype within the same generation. This is actually pretty prominent. One example is the case of Candida albicans, which is a fungus, which switches between white and opaque phenotypes. It in fact does a lot more, but for now let's just consider this switching. And it can switch between a very virulent state and a mating state in order, depending on the environment and so forth. But the fact is, its switching is stochastic. So you can, this is the kind of schematic that we have. It does not have all of the resources necessary to get exactly what its environment is like. So it just has a certain rate by which it switches from one phenotype to another. And so we wanted to see in those cases how exactly would the organism grow, because that will help us do things like antifungal treatments for this kind of pathogen.
Okay, so we can think about the problem in a Wright-Fisher way, where we have a population with a wild type and a mutant that can have two possible different phenotypes, A and B, and they both have a specific fitness that is relatively either plus S or minus S with respect to the wild type. And then we can make them reproduce with the fitness, and then there is a part that comes with the phenotype inheritance, that it's stochastic, and there is a switching probability with respect to your parent that comes with new. And then what we want to really compute is the fixation probability in order to somehow try to understand how if there is an evolutionary advantage in having a switching phenotype. And this one we can do it on top of a switching environment that switch with the probability epsilon, and what really does is just switches the sign of the fitnesses in our mutant. So we can simulate the Wright-Fisher model first, thinking about the static case. And here you can see a plot where we have like the frequencies of the two phenotypes, A and B. Every single red line is a path, a Wright-Fisher path, and the blue line is the absorbing boundary of fixation, where the two frequencies sum up to 1. And what was really surprising to see is that the population is able to calibrate really quickly and it keeps the ratio constant. So we were thinking about whether we could use it as an effective lower dimensional dynamics and exploit it for an heuristic analysis that Alex is going to talk about.
Okay, so on the right hand side you can see the numerical results for the fixation probability as a function of the switching probability when you start with a good phenotype with 3 different selection coefficients S. And what you see basically is just that the fixation probability decreases with the switching probability, which makes sense because with a higher switching probability you have more bad phenotypes within the mutant population, and this will lower the mean fitness, which decreases the fixation probability. So in order to get a better understanding of this, we try to find an analytical approximation. And so if we only have 2 types, the White-Fisher fixation probability is given by this equation, and we can compute it by knowing the population size and the selection coefficient between both types. Now, but here we have 3 types, and the idea is to treat the mutant population as one type, and then we have to compute the mean fitness of the mutant population. So the only thing we have to do is to solve the set of equations which gives the frequency of the good phenotype and bad phenotype for its stationary state, also called mutation selection balance. And from that we get the mean fitness, the stationary mean fitness of the mutant population. And now we only have to subtract the fitness of the wild type, which is 1, to get the mean selection coefficient. And now we plug it in the fixation probability, and now the dashed line is the heuristic approximation, which fits quite well. Now the more interesting case is when you start with a bad phenotype. Now you want to have a switching probability which is different to 0, because otherwise you don't produce any good phenotypes. And then most likely the population will die. But also you don't want to have the switching probability to be too high, because this would decrease the mean fitness, and through that the fixation probability. So also here we try to find an heuristical approximation, and the idea is to compute the number of good phenotypes that can occur before the bad phenotype population goes extinct. And this is basically done here in the first line, and we make some approximations. So for example, we approximate that in each generation only one switch can occur, which makes sense if nu is very small. And also our mean population, the bad phenotype population is very small from the beginning, and then will only constantly decrease. So from that we get this prefactor, which we then just multiply with our fixation probability for the good phenotype. And then we get these approximations, which seem to work quite well. And if you are not convinced by this, we also produce much more data and put this on a log scale for both cases, and we see a very good agreement. So what we also did was we rescaled for the bad phenotype case the fixation probability and the switching probability by S, and then we can see that the behavior basically depends on the ratio, and that the peak occurs when nu is equal to S.
So this is basically telling you the optimal value of nu and S, right? To pick what's the optimal switching rate nu to increase your probability of fixation. Let's see. Okay. So you can write down the Langevin equations for this. So we have the 2 phenotypes, XA and XB. XA is the good phenotype, XB is the bad phenotype. And they have the setup here is symmetric, so their fitness effects are plus S and minus S relative to the wild type. These are the fitness advantages and disadvantages relative to the wild type. And the dynamics happens in this 2-dimensional phase space. And the idea is that we start at frequency of 1 over n, starting from either one good phenotype or one individual bad phenotype. And we're trying to figure out what's the probability of fixation. That is, what is the first passage? It's basically a first passage problem to reach that line where XA plus XB is 1, because that means the mutant population has actually fixed. And so if you're clever, like Federica and Julia, then you would be able to actually figure out that the right change variable is the sum, and the relative asymmetry between the two phenotypes. So that would be the difference divided by the sum. And if you do that, nice things come out. And so these are the Langevin equations. This is the sum dynamics, and it looks like it's carrying out this, it has this time-dependent effective fitness, which is coming from this asymmetric variable, which is this difference divided by the sum. And the other nicety is that obviously if these are the right variables, your covariance matrix diagonalizes.
Okay, so basically this is, as he said before, an effective equation for a single allele that tries to invade the wild type with an effective selection factor which is S times WT. But the problem with this selection factor is that it is stochastic and time dependent, so if we want to look at its full evolution, it is coupled to Y, but we can get rid of this dependence on Y if we consider this as a deterministic variable, only as regards W. I mean, we drop the stochasticity only for W, and we find an ordinary differential equation which is actually solvable. If we look at the fixed point, the physical one, it is the same as the prefactor that previously Alex found by imposing the mutation selection balance inside the mutant subpopulation. And what we did before was plug in this value here, this value here. Okay. It was plugging this value here into Kimura's formula, and we saw that it did a pretty good job for the case where we start with a good mutant, whereas we need corrections for small nu if we start from the bad mutant. So why does it work so well? Is the Langevin, are the Langevin equations giving some insight? And the answer is yes, if we consider timescales, because this approximation basically consists in taking the asymptotic value, so there is no stochasticity, as we said before, but also no time dependence. And this is good only if we can make an adiabatic approximation and consider Y as the slow variable and W as the fast variable. So the picture is the following. We can start with either the good phenotype or the bad phenotype, and as we go on, the size of the population, of the mutant subpopulation increases, but there is also relaxation of the composition of this subpopulation to the equilibrium value. If this relaxation is fast, this approximation holds, and this is the case if we start from the good phenotype, whereas if if this is not so fast compared to the increase in size of the mutant subpopulation, then this approximation is not so good and we need corrections. And we tried to look at it in the data. We found, good agreement with this prediction, and we also managed to compare the simulations with the analytics. Here you have the case for big nu, which means frequent switches, and in this case they relax pretty fastly to the asymptotic value. In the other case it takes a little bit longer, but we see that there is an asymmetry between the start with the good mutant and the bad mutant, because if mu changes, if mu is very small, the asymptotic value is very close to the initial condition of the good phenotype.
Okay, so now we're going to change gears a bit and start talking about some numerical results for the case of the switching environment. After each generation, there's some probability epsilon that the environment switches, and the fitness parameter S flips its sign. That environmental switching probability is epsilon. Here I've plotted the fixation probability as a function of the environmental switching probability on the x-axis and the mutation probability, or phenotypic switching, on the y-axis. If you start with the good phenotype, initially the one that matches the environment, it turns out that increasing the mutation rate or if the environment changes more rapidly, both reduce the fixation probability. The reason is that the population is always going to initially grow, and then when you start having mutations or the environment flips, you're going to lose a significant fraction to selection. Things are more interesting when you consider when you start with the bad phenotype. if you consider small epsilon, you recover the case we saw before, you kind of have this peak with the optimal epsilon. If you consider fixing nu small and you start increasing it, you actually see a small peak here. We kind of heuristically understand that as it kind of performs the same effect. So the bad phenotype only really grows when there's a mixed population. And so this, in the absence of mutations, the environment switching can help it grow, because then it becomes the good phenotype. And so like the curves you saw before, what happens is the small nu region actually starts to lift up. But then as you increase epsilon, but then once you pass a certain regime where nu equals epsilon right here, it starts to drop off because the phenotype switching can't keep up with the environment.
Okay, so I'll try to keep this fast. How do we switch? Okay, so here we're looking at the effect of the switching environment on the fixation probability of the deleterious mutant. And so on the x and y axes I have plotted S, we're sweeping over S and nu here for a given epsilon, which again is the rate of the environment switching. And the color indicates the difference between the fixation probability of the mutant and the switching environment versus the fixation probability of that mutant in the static case, the static environment. And so for slow switching of the environment, we see that the interesting behavior is for occurring at high S, where you have an increase in the fixation probability. And our reasoning behind this is that if you have a high enough S, although that's extremely deleterious at first, if you have a finite nu, you'll switch the good mutant, and you'll have a higher probability of then fixing. So this kind of makes sense. Whereas if you have now higher or faster switching of the environment, the red region starts to disappear and this blue region starts to arise. And this is because your environment switching so fast that your mutant's effectively neutral. And so your fixation probability actually decreases in the switching environment as opposed to when you're just in the static environment. So there's plenty of other features to go into this plot that we would like to look into, but I'm going to move forward here. So this is just a sample trajectory of this red region picked out from one of this red regions. And so the red curve is the tracking the deleterious mutant in the population, and the green curve is the good mutant in the population. And like I said, we see that initially, and it's important to note that we started with the deleterious mutant here. We see some switching going to the good mutant, and that starts to increase, but it's also important to note that the deleterious mutant starts to follow, and like Michael just said, it seems that the best strategy is to actually have both phenotypes kind of around, because in the case of when the environment suddenly switches, you don't just die, because if you put all your eggs in one basket, you're going to go extinct as soon as the environment switches. So, and what we would like to do moving forward is adapt the heuristic solution in the static simple case to this more complicated case of when you have a switching environment. And we want to just thank everybody for listening. Thank you. Any questions?
Kind of a basic question I get, but for your example at the start of it was a fungus, I believe.
Yep.
Was it clear which state is the more fit and which is the least fit?
Depends on the environment. So one of them is the virulent state and one of them is the reproductive state. So depending on what the environment is, it will want to switch between the two. In that particular case, actually, it can sense some things about the environment, so it sets the switching rate, but it doesn't know exactly what the environment is. And there's another paper by Kussel and Leibler where they discuss when it's more beneficial for you to have as many stochastic switching versus sensing the environment, because sensing the environment can be pretty expensive itself for an organism as small as that.
Okay, and if I can ask another question.
Yeah.
It wasn't clear to me why you wouldn't just have one half of the population having one phenotype. I mean, what did you, like, maybe I missed the point entirely, but what does the switching give you other than having two phenotypes and they never switch?
Well, in that case, if you only have one half in that way, then when you have the deleterious one, then so suppose you over there you had white and opaque, okay? Now suppose you enter the situation where it's very bad for the white. Then the white will go out almost completely. And so then later on when you enter the situation where it's beneficial to be white, you don't have any of the whites left.
Okay, we will thank this group and we'll take no more questions because we're out of time, and the next group should come out.
I think he's drawing something. Can people hear me? Okay. Okay, so we're group 10. Hello. So basically our project boils down to trying to link or establish a link between the brain of an animal and its behavior. So you can imagine if you take an animal, its brain controls its behavior, but its behavior can also influence the brain. So if I punch myself in the face, like that's me controlling my behavior, but then I'm not going to do it again because it hurts. So my behavior has then influenced my brain. So that's just an example. But so yeah, so you got this interplay. And we just want to try and, establish some sort of link between these two things, the brain and the behavior. So the way we want to do this is to basically say, okay, let's treat it like a dynamical systems problem. So the state of the brain we'll call X. That could be like neural activity at a given time. And then you also have the behavioral states. That might be, say, the positions and momenta of the limbs of an animal or something like that. And then what we're going to do is we're going to write down two coupled differential equations for x and y, which are here. So x dot is just some function of x, y, and time, ditto for y dot. So that's all well and good. Basically, what we want to do is find f and g. Okay, so we then have to choose an animal. So animals generally are very complex things. So like the human brain has 100 billion neurons or something crazy like that. So this is why we choose C. elegans, which is this thing which you've seen a few times during the school. It's very simple. It's 302 neurons, which is great if you're a theoretician. And it's also transparent, so you can actually monitor its brain activity and its behavior simultaneously if you're an experimentalist. And that's been done. So we have data, and that's what we're working with.
Okay, so before we started looking at the data that we have, we wanted to start with a system that we knew to kind of check to see if our methods were working. So we started with the Lorentz system. It's a system that we all know how the dynamics work, given different parameter values. And so we started with that and then moved on to, we have some data from, I believe it's the Leifer lab at Princeton, where basically one of the C. elegans worms is crawling around on a dish. We have time series of neural data, which is the fluorescence response to a calcium concentration, and then also the behavioral data, which is the different eigenworms through time. So to start with our methods, I'll hand it over to Pansy.
In our big group, we did two different approaches, and parametric and non-parametric, and I think you all have heard about so many like machine learning models throughout the school, but I put this definition here, you can read it, and I think what I think about parametric approach is that you have a bunch of function forms you choose, and then you learn the coefficients of the function forms through the testing data. So for parametric approach, we use the model called CINDI. The acronym is CINDI, but the long form is Sparse Identification of Nonlinear Dynamical System. It is quite a robust method, and in the paper they talk about like linear, nonlinear oscillator, and they talk about turbulent flow against the obstacle, how they got these equations from these dynamical system out of the measurement. Unfortunately, it failed in this system, but we tried it, and I would like to show you my attempt. And before I go on to my data, I would like to talk more about CINDI system. Is that when you have X, Y, and Z, you choose a bunch of functions here. You calculate it, and you have also X dot, Y dot, and Z dot. And from here, you can calculate the coefficient here. And from the name, you can find it's sparse. So the idea is based on this coefficient. You looked at the coefficients which are below the threshold, and you take, if let's say like this coefficient is really little, we take out that function, like respond to that coefficient. So we make it sparse, and we keep removing the potential function, and we ended up with a dynamical system. So, but we have to note that the assumption holds only for the system with suitable basis. As my group already says, that we want to find a dynamical system between the behavior and the neurons, but unfortunately we couldn't find the derivatives in the noisy data. Actually, we can use a total variational regularization method to take the derivative in the noisy data, but we didn't use that. So we used the time derivative data in the data set. And we looked at these three neurons, which are neuron 35, neuron 30, and neuron 18. How can I choose these neurons is that my group calculated the mutual information for these neurons, and this is the dynamical system I would like to find, and I couldn't find. These are the bases I use for the Lorentz system. I mean, you know that I a little bit cheated on that because these functions are in the Lorentz equation. So I use the same basis, and I calculated here, and I have data and the prediction, but it didn't work. And you can see that why it didn't work. Actually, my another group partner, Adelaide, calculated using the different bases, and she used the polynomial up to the order of 5, including all the combination of X, Y, and Z. And in Lorentz system, you can see the test data and predictions are on top of each other, and we are able to find the dynamics of the system. And when we plot the prediction and the data, it goes in a line. And but she used a different basis function as me, but she still got the really weird behavior here. So the fitting is really bad. So why CINDI didn't work? Probably because of the choice of the basis function. Probably because of the sampling error between data and the time derivative of the data. And also the CINDI model based on the assumption that this dynamical system will be first order. And I think it might not be the first order differential equation. And however, Lorentz system work with CINDI because it fit all the above requirements.
So, no. I can stop, and then you will do it. So again, our aim is to find something like this. But however, in general, especially with neurons and with eigenmore, we don't have so many measurements. We have something that looks more like this. So this is like the X time trace. So the idea is from something that looks like that to reconstruct, in some sense, something that is similar to the Lorentz system. So we need to find a method that gets us from here to this, and this is actually the results of our method. So this is like the two principal component projection, and this is in 3. You don't see very well, but I mean it's in quite a good like relationship with the Lorentz system.
Now we need a time series, and for our data we can start either from one single neurons or one eigenvalue or mix of them. Then what we want to do is to find some kind of phase space, which we don't really know what it means, but it should be homomorphic to the real phase space, and in this sense we see that the 3D phase space meets that requirement. However, if we have only this dynamic, so we measure only the X, for example, for Lorentz system, nobody's telling us that it's 3-dimensional, it could be 50-dimensional. So the basic assumption of this is that there exists a low-dimensional dynamics that governs our system. And these are the results. So in our method, what we do is that we calculate the mutual information, and it's a plot there. And from this plot, we can say that, okay, for the Lorentz system, this is like around 15, it's a good threshold. And then we build a matrix like this with all the various time trees. So the time t, t plus 1, t plus tau, and then we go down and we shift it of 1, and we do again. And from this matrix, we do the SVD. In the SVD space of d dimension, we take a ball of points. And we see when this ball will completely diverge into the attractor, and this is the plot here. So this is in some sense the radius of the ball, and the fact it's the log of the radius. So, and the fact that this is linear tells you that the system is chaotic and there is some Lyapunov exponent. And from this plot here, we can find if we measure. So the curve is something like this. So it goes straight and then it goes to a plateau. So if we take this area here, this in some sense tells us how long the system stays predictable in that kind of dimension, in that number of dimensions. And this is exactly what is plotted here. So this area is in some sense a proxy for the prediction accuracy. And we know that the Lorentz attractor has 2 point something dimension. So you see in this plot that 2 is already very high, but 3 is better. And then if you take more dimensions, you will fall off into the noise. And so you go down. And so this is a wonderful prediction. So we see that clearly this has 3 dimensions. However, in order, the starting point for this is the mutual information, and we need to calculate this mutual information somehow.
All right. So so before basically to study the neurons and eigenworms, basically you take the system, you try to find dynamical equations. That's the parametric approach. The other ways you could use information. So in the parametric approach, you can basically find a relationship between how one variable may affect others, how a neuron may affect an eigenworm. And so you can also use mutual information to do that. So all of you are probably experts in mutual information. When I came in, I didn't really know anything too much about it, so I had to come with a toy example just to kind of figure out what was going on. So this is just a really simple system, R1, R2, and they're basically coupled through this parameter, a stochastic system, which is coupled through this parameter, which I call alpha. And this is kind of cool because you can actually you can find exact probability distributions, and you can compute exact mutual information. Of course, if you have experimental data, you don't have equations, you just have a bunch of points. So to actually numerically compute mutual information, you should bin your data, essentially. And so the exact answer is basically the orange curve, and there's a few things to get out, which is that if the coupling is 0, the 2 distributions are independent, so you get 0 information, and then as you increase it, you should basically get more information out of the system. And the points here are basically sort of what I got from my code, basically to compute the mutual information, and of course what you find is that there is an issue with how you choose the binning and how you choose the clustering. We weren't able to resolve this at the time, but that is something to consider. But you still should see the features. And so, given the time traces, you can basically take, for example, all the neurons, take a neuron and compute the mutual information with one of the eigenworms. And so this is basically each eigenworm computed against every neuron. And just the key thing to get out here is there's a few spikes that you see. And so, for example, for eigenworm 3, which I'll show in the next slide. Neurons 18 and 30 have, basically the largest spike in mutual information. And so if you look at the actual time traces, you basically find that indeed whenever eigenworm 3, say, drops, there's a spike or a drop in the neuron 18 or 30. If you look at neuron 12, which is basically the lowest point here, it looks essentially just noisy and there's no correlation.
Okay, so this slide we can skip. But this one is actually crucial. Because here we see again the prediction accuracy in function of dimension. And we see that both for the one neuron that we see at the most spike and for one eigenworm, the dimension is 3. And this tells us that we cannot in any possible way approach a neuron thinking it's a one-dimensional system. We need to have more than one. We need to have at least the 2 inputs, or maybe these 2 inputs are like linear combination of something else. So this tells us that, okay, if we want to study a neuron, we need to take into account something more than just the single neuron time trace. There is something else. There are some other objects that are contributing to this. And especially for this neuron, this is completely clear that we are at all not in a one-dimensional system.
So then we ask the question whether we can predict the future from the time series. Right. So the strategy we are going to take is to the 80% to 20% split of training to test. And we're going to train a model to the training time series. And we'll train a model in a two bases. One is this original time space, and the other is reconstructed phase space from Lorenzo. And we'll predict the time series from that and see how it fares against the test data. So first off, on the original time trace, well, as you can see, the prediction failed miserably, but this is not so surprising. Oh, by the way, I used the autoregressive model, which is the common method used for time series prediction. Basically, you are using p previous time points to predict the next time point. And here I used from 5 to 40 p with pretty much the same result. And it didn't work because, as we have shown before, we believe the neuron is governed by nonlinear dynamics. And from previous slides, the embedding dimension here is 3. So it's actually governed by a third-order differential equation. So we went on to that reconstructed phase space where we have 3 phase space variables. And we applied the Cindy method again using those 3 phase spaces. And so we trained the model and we predicted the derivative of those phase spaces variable. And as you can see on the left, the prediction was pretty good for first and second principal modes. And since we have a nice derivative, we simulated the time trajectory starting from the last point of the training data. And as you can see on the right, so our simulated trajectory closely followed the actual trajectory in the phase space. So we believe now we have a model that describes the dynamics of this neuron in the phase space. And next is we project that back into the original time space, and lo and behold, that projected time traces closely followed the actual test time trace again. So basically we were able to see the future. And the moral of story here is we basically use the same model first onto the original time series, then onto the reconstructed phase space in 3 dimensions. First time it didn't work because we are trying to push the linear model to the third order, to the dynamics that's governed by third order differential equation. But when we reconstructed the phase space, we nicely resolved those nonlinear dynamics into the linear dynamics, and we were able to predict the time sequence. Yep. Thank you.
So when you talked about the Cindy method, I have no experience in dynamical stuff. So you said it didn't work and you showed something, but I don't know what it was supposed to look if it worked. So what did you expect? And
you can see here that in Lorentz system it works, so you can see the straight line here between the function like f of x, y, and z and x dot. So I'm supposed to see a straight line here.
Oh, I see. Okay.
So I was wondering, since this worm has only 3 neurons, like if there is any, if it's possible to maybe block one of the 3 and try to block it, like destroy it or something like that, and try to reproduce the same experiment. And
you said a worm has 3 neurons?
Yeah, that's what
We are so sorry about that. It's not 3 neuron. I just choose the 3 dominant one based on the neutral information calculation. I don't really know what will happen if you plot one of those.
So I'm just wondering, like, about the dimension of, like, why the dimension is 3 and...
Well,
yeah.
I mean, in general, I would say that, okay, if you block one, you will not see the same dynamic. So maybe the dimension will be 2. But I am not at all sure that the effective dimension there is like neuron eigenworm, and it's not like 3 like very heavy linear combination of things that could give rise to anything else.
So yeah, I was wondering if it always tries to maximize like the computation power that it has.
I don't know. I mean, I don't know what happens if you block one. I know that some are 3, some are 4, so it's not always 3.
Okay.
Okay, I think we're out of time for this group, but thank you.
Okay, now the last group.
Not least,
Yes,
group 3.
exactly.
So we are going to present our project on the dissipation in models of flocking, and especially we will focus on the VCheck model. So as a short introduction, the VCheck model is a theoretical model that describes active matter and collective motions in flock. So it is a non-equilibrium phenomenon, since every element in the flock they dissipate energy during their flight, and in order to perform a systematic motion. So there is a phase transition between ordered and disordered system, and there exists some theoretical framework that describes the emerging collective behavior, but none of them use thermodynamic quantities such as entropy production in order to explain this phase transition. So that is basically the goal of this project. So the VCheck model, it's a model where each bird will reorient its flight direction in the time depending on its neighbor orientation. And so in the implementation, in between 2 consecutive time steps, there is a first step where each bird reorient its flight. So this orientation is made according to 2 terms. The first one is a deterministic one, where which is a weighted sum of all the neighboring orientations, where the weight, Jij, exponentially decreases with the distance to the bird. And a second term, which is probabilistic, with a noise eta, which is a Gaussian distribution in all the directions of space. And then once each bird has a new orientation, they fly to the new position at time step t plus the delta t. So in order to characterize the state of the system, we will use principally 3 quantities. The first one is polarization, which is, okay, I had changed, but okay, no, that doesn't matter. Which is not that, but which is the norm of a vector, and each of the component is the average of the corresponding component of all the birds. And when it's ordered, the polarization tends to be 1, and in the disordered phase it goes to 0. There is also the entropy prediction, which will be discussed in the next slide.
Yeah, thanks for introducing the model. So now that we know what we're talking about, of course, everybody's probably waiting for movies, so that's going to be this slide. If it plays... Come on. No! Okay, how do I get this to play? I probably won't. That's super sad. Okay. So unfortunately no movies. Okay, so I mean what you would be able to see here is that we initialize the birds randomly, and then they usually aggregate in these sort of swarms that then very mesmerizingly fly across the screen. And what you can imagine there is that's a decrease in entropy because the system becomes more ordered. And we actually do see that in these entropy rates we calculate, but that's not what we're interested in. So here on the right you can see that the model looks a bit more ordered because all the birds fly in the same direction, which you don't see because it's not moving. But that's the state we're interested in because that's the steady state. So looking, okay, where does the, okay. Then, okay, so now the question is how do we actually calculate this entropy production? Most of you might have seen this formula before. Basically what happens here is that we look at single trajectories of the system through its state space, and then the entropy production has a sort of obvious contribution, which is basically the difference in entropy of the final and the initial state. And then in addition to that, we get this non-equilibrium contribution over here, which is due to broken detailed balance in the system. And so this is the part that we're interested in because that's the entropy production due to activity, whereas this thing in steady state should vanish because the initial and final states, if we just average over a steady state ensemble, should be the same. And to kind of give you an intuition for that, if we take the time dimension out of this model for the time being, we can basically just write down a Markov chain with unbalanced transition probabilities. And as you can imagine, this thing in steady state just cycles. And that's basically what we try to measure with this entropy production. Now, getting the time dimension back into the system, we try to think about, okay, what are sort of our states in the system? Because the problem is that these birds have velocities, so you can't just think about forward and backward rates, but you have to think about time reversal. And then the question is, how do you incorporate that with velocities? So we came up with a kind of nice idea to sort of incorporate redundant information into our state variable, namely the velocities that take you out of a state or out of a positional configuration, and also the velocities that take you into that. Because then it's easy to write down a time reversal operator. You can just imagine these velocities switch and get minus signs, and we can incorporate the equation of motion into an evolution operator, and then we can immediately write down these forward and backward transition probabilities and implement that in our model, which should be the next part.
Cool. All right, so now that we had the entropy production, we knew how to calculate entropy production, we started writing down simulations. And the first thing we looked for was to see that we were getting the phase transition that we knew we should be seeing, which is between the disordered and ordered phases for these flocks. And so you see when we have low temperature, and when the interaction strength between the birds is higher, we get an ordered phase, and when the noise is larger, you don't get an ordered phase. This also all sort of collapses if we rescale, if you look on the right. So then we can calculate entropy production, and we can relate that to the phase transition. There's a couple of limits here that should be somewhat intuitive. So at really large noise or really high temperature, you basically, none of these birds are not really interacting with each other. The noise totally dominates. And so you essentially just have non-interacting particles moving, and so you should have no entropy production. And then all the way at the other limit, so if we'd extended this temperature axis way this way, and if we do extend it, you see that this decreases very, very slowly. You also get zero entropy production, because if you have a polarized group of birds that are all not moving relative to each other, you should get an entropy production. The entropy production comes from the changes in the JIJ matrix at each time step. So this is what entropy production looks like for 128 birds simulated together. And we can check how this goes if we change n. So these are the polarization curves, and this is for three different sizes of the bird flocks. And I don't know, there might be a difference there, but we can't tell. But yeah, Jordan?
Okay. So I'm going to quickly describe what went wrong. How do I go to the next? So this is, sorry, whoops. Oh, shit. I think it's good that this Sorry about that. So this is delta t. So initially, we were running our simulations with a relatively large time step, and we were getting this very exciting large peak in the entropy production at the phase transition, which we thought was super interesting. But it turns out as we cranked down the time step, we sort of approached this sort of like stable lack of a peak, I guess, which is actually a relief because, so this is in 2 dimensions. In 3 dimensions, we were getting a negative peak. So the system was absorbing entropy at the phase transition. But that too, as you crank down the time step, seems to go away. So, but I mean, we had pretty poor statistics here. But if we continue this, we want to run more. But yeah, on to other things.
Okay. As Jordan was producing these beautiful plots and awesome movies that you couldn't see, in parallel we were also thinking, okay, we have polarization and entropy production rate to characterize what happens in 2 different phases, but can we look more closely into these phases and see actually what happens to each bird as they move and align with each other or be kicked around with these random noises? So I think one of the obvious choices to do this is to find the correlation functions, and these are the 3 examples. It might not be the most optimal one, but that we tried for now. So first one is the spatial correlation. So if we want to find the C of R, at certain time point, we just find 2 birds that are R apart from each other and take the cosine of the angle between those 2 velocity vectors, and we also find the autocorrelation vector, autocorrelation of each velocity vector. So we wait for delta t and take the dot product, and then we can also do the autocorrelation for every pair of the birds. So in this picture, we just take I and j, which is like red and blue birds, and there should be an initial angle at time t, and then we wait a little bit, and the angle between those 2 vectors should change, and we just find how much it changed, and that could depend on the noise or the alignment that happens between the birds, and we take all the average. So this is the preliminary results. So these are all for 2D and just a single choice of dt, and probably we should optimize the code to explore other regimes as well. But in general, we see similar type of behavior. So at low temperature, at any temperature, we start from 1 just because you're taking dot product with yourself. So and then you have a very small decay as you increase the distance in terms of space or time, and as you increase the temperature, you see this decay rate start increasing, and then if you go up to really high temperature, now if you have a small finite change of distance or time, you almost immediately go to 0 up to the resolution we could probe in our simulation, and now you just have flat thing because the birds cannot align with each other. They just lost all the correlation. So for each of these curves, we fit to the exponentially decaying model, and then we plotted the k's, and this is what we see. So after it decays to super fast, we can't really fit it, but as you can see, it's kind of consistent with what we see from the polarization and the entropy production rate. So you see this gradual increase of this decay rate, which shows that if you're really well aligned, you won't be decaying really fast, so you're in the aligned state or order state, and there's some second order transition to disorder state where you completely lose order and you just move randomly, and now you're not really producing any entropy because you can't really align with each other.
So having somewhat established the existence of entropy production in this model, we decided to try some analytical calculations to give us more insight into where the entropy was coming from. And so what we did was we tried calculating the forward and backward probabilities for a short time or one time step. And so what we're doing here is that we're starting from an initial state where the velocities of all the birds are given by ui, and we go forward in time by one time step, and we end up in a state w. And this probability is just the probability of the noise that is required to get from u to w. And then similarly, we can calculate the probability for the reverse trajectory. So you start off at minus w and you end up at minus u, because the signs of the velocities are flipped when you reverse time. So what we obtained after this, so having obtained this ratio for one time step, we just multiplied those values over an entire trajectory, and then we took the continuum limit where we set delta t to 0. And after about 20 pages of algebra, what we found was that the entropy production is made up of these two terms. So the first term, which involves a time integral over the trajectory, is analogous to the term we found in the earlier slides, which had sums over intermediate times. And this second term is just the entropy of the initial and final states. So what this tells us is that because this first term involves a time derivative of the interaction, this tells us that the presence of network rearrangements, which means that the birds keep changing their neighbors, that is what leads to the production of entropy, and that makes sense because that's what you would expect. Also, this is also nice because it shows that if network rearrangements are absent or JIGs are constant, that means that there is no entropy production, which again is what we would expect. But there is one problem with this formula, and that is that it only works for metric interactions, because if you have topological interactions, the JIJs are not continuous, and so the time derivatives don't exist. And because it took us a while to figure all this out, we weren't really able to compare this formula with numerical simulations, but I think that's the logical next step. So to conclude, we've calculated numerically the entropy production in the VCheck model in 2D and 3D. We've studied the phase transition between the ordered and disordered states, and also the spatial and temporal correlations between birds. And we have managed to do an analytical calculation of entropy production, but like I said, it really does need to be compared with our simulations.
So just quickly, one of the, of course, sort of these dirty secrets of the VCheck model is if you don't impose toroidal boundary conditions, the whole thing falls apart, right? So how do you think, if you were to put this in a non-torus, so just in an open field, right? Because usually you would need some sort of like a coalescence or some sort of a long-range attraction term in order to keep this thing together. How do you think your results would change, or do you have any intuition for that?
So I think some of us did try changing the system size while keeping the number density constant, and we didn't really see any effect, which...
Well, yeah, right, because the number density is constant. It's more just there's no... It's more just because you don't come back around across the edge. So you just sort of float off into the nothingness.
Right. So in that case, I'd expect entropy production to go to 0 at some point, because I mean everything just disperses, right? So would be weird if you still.
You could add a coherence term.
Sorry. Yeah, okay. I honestly have no intuition for what's going on there, but I imagine it would look sort of similar because effectively you just need something to confine your system, right? So you might also just try rigid boundary conditions or something like that.
So in the plot where you showed the entropy production as the transition happens with temperature, there's like a shift in temperature between when the transition happens, where you see it in the polarization, and the entropy production there. Do you know what the shift is, or?
Wait, what shift? Which shift?
Like in temperature, you see like the slope. Yeah.
Oh, those are just, I mean, one. Yeah, you want.
I mean, they're on totally different scales, right?
The x-axis
no,
is
on
a blue curve, half height size at different points.
Right.
I think it kind of matches. I don't know.
Wait. There. Temperature is factor of 2 or 3 different
Right.
transition points.
Yeah, I don't think we know. I mean, it could be this is a relatively small system, so maybe it's some sort of finite size thing. Yeah.
Okay, so did you or can you look at like the entropy production like density as a function of, say, the space? Because there's like a work a while back that said, so at least for active particles, not the VICSEC model, but the entropy production is maximized sort of at the boundary of these clusters. So like if you take a flock, where would the entropy production be maximized?
Yeah, so we actually looked at that at some point because this formula that we wrote down is a sum over all the birds. So you basically get entropy production for every single bird. But you don't have anything you can average over if you look at single birds. So the whole thing is just super noisy because basically what you do is you draw two random variables from two slightly different distributions, and then you just ask, okay, which one is more likely? And since you only draw once, it's just super noisy. We tried to average that a little bit because it would give super nice movies or something, but we didn't find anything conclusive. Maybe that's something worth exploring.
But also you're asking about effects at boundaries. Like back to Gordon's question, there aren't any boundaries in this system. Yeah, but the whole flock is basically goes all the way around a torus.
So let me ask also a question. There is also additional term in the entropy production here, which is how do birds actually measure the velocity of another bird? Right? What do you think is contribution of that? Have you thought about that? Right?
So the short answer is we haven't thought about it. The slightly longer answer is, so I'm not exactly sure which term you're referring to. So I mean, also the birds are actively flying and therefore like burning energy and generating entropy, which is also something we just don't consider in this model because we just assume constant velocity. Does that sort of go into the direction of what you're asking? Okay. Yeah, Liam.
Okay.
Also, if you were to consider an extension like Gordon talked about earlier, if you had some sort of momentum term, imagine that this would have an effect on the entropy production rate.
What do you mean by momentum term?
So that, like, I imagine that the reversibility of the trajectories, I mean, they would be more reversible in time.
So we're not implementing this model precisely such that like at every time step something completely rearranges. There is a tendency to just keep going straight at any particular
there is.
step. There is an inertia here.
Sorry. Okay.
Because as you take DT small, you get very funny results if you don't have that kind of thing.
Okay. I think we're going to end.
Yeah, we should thank this group again.
Thanks. I think we are done pretty much. So the school is over.
I'm done.
Yes, we're done. Go have fun. And one last exercise which I wanted to give you is, remember that the 2007 school, biophysics school here, Alex and Thierry were students here, and then they became organizers in 2019, so it's 12 years down the road. So think about which ones of you, and maybe vote between yourselves, choose who is going to be organizing the 2031 school, okay? So it's the last exercise.
I wanted to